There's a running joke in med school where if some professor asks you what the number one risk factor for a disease is and you have no idea, the best bet is just to say age. It's not universal, but age really is the common thing underlying most chronic diseases. Cancer, cardiovascular disease, liver and kidney problems, all of these increase with age. And then, of course, there's dementia, the archetype of the age-related chronic disease, and the one many of us fear the most due to the lack of adequate treatment. But what is age? I mean, really, why should the number of times you've circled around the sun make any difference at all? Is there some cellular clock inside us ticking away incessantly, irredeemably, like the telltale heart? Is the rate at which we age fixed by genetics, or is it changeable? Can we slow it down? And if we could, would those age-related diseases, if not disappear, regress into the distance? These questions have led to the concept of biological age, the idea that by measuring something, and that thing differs depending on who you ask, we can assign a more accurate value to your age than the number of candles that appeared on your last birthday cake. And that's cool for two reasons. First, because it would allow us to better estimate your risk of age-related disease. But second, because unlike calendar age, biological age can be changed. I'm wondering how old I really am this week, thanks to this article appearing in the journal Neurology, which examined the risk of dementia based on biological, not calendar age. Let me walk you through it. The population studied were people who contributed to the UK Biobank. This is an absolutely huge undertaking containing medical information, genetics, and biomarker data on more than 500,000 UK residents. Researchers identified over 280,000 individuals in the biobank who had adequate data to calculate their biologic ages and who had available genetic data and who did not have dementia when they joined the study. So wait a sec, how do you calculate biological age? It depends who you ask. Some folks use DNA methylation, or so-called epigenetic clocks. Others use more readily available measurements, things like blood pressure and lipid profiles, to estimate age by so-called phenotypic clocks. You can even use echocardiogram data, according to one study. This is actually one of my problems with biological age research. Any researcher can basically take a set of measurements that correlate with age, say hair density, and then claim they have created a clock based on that relationship. Sure, I'm 45, but my hair age is 53. In the case of the neurology study, two well-validated clock equations were used. One is called pheno-age, which integrates information from 9 blood biomarkers, and the other is the Clamara-Duval biological age, which includes some other measures, as you can see here. And just to harken back to my prior point about the problem with biological age clocks, even in this study, these two biological clock measures, which actually share quite a few parameters, didn't agree that much with each other. They had a weak correlation coefficient of 0.29. And the authors don't give us a scatter plot to visualize that, but here is what a correlation of 0.29 looks like, just to give you an idea. This is just something to keep in mind when someone tells you, or worse, tries to sell you a test to measure your biological age. It's not like there's a gold standard here. Of course, it may be that these two clocks are capturing different aspects of aging. Sure, my hair age might be 53, but my pancreas age is a healthy 41. Anyway, the interesting question is whether either of these clocks predict the risk of dementia. Over more than 10 years of follow-up, 4,770 individuals had a new dementia diagnosis, and as it turns out, older biological age, no matter which clock you use, was a significant risk factor for dementia development. But I should clarify something here, actually. It's not surprising that older biological age is a risk factor for dementia, since one of the factors that go into the biological age calculators is calendar age. So take a look. I input some normalish lab values into this pheno-age calculator along with my birthday. I'm 45. My estimated biological age is 39. Nice. Then I left everything the same, but made my birthday 10 years later, so my calendar age is 35. My calculated biological age is now 30, 10 years younger. So therefore, what's interesting is not really your calculated biological age, it's the difference between your biological age and real age. Like, are you aging faster or slower than expected? This is where the rubber meets the road when it comes to dementia risk. And the answer again is that yes, if your biological age is older than your calendar age, your risk of dementia goes up by about 1% for each year. So if my calendar age is 45, but my biological age is 55, I have around a 10% higher risk of dementia than I would if my biological age matched my calendar age. And this held true for both measures of biological age. The effect was dramatically magnified in those with a key dementia risk factor, the ApoE4 gene. This is the primary genetic risk for Alzheimer's disease. Among ApoE4 carriers, the 25% of people with the greatest difference in biological versus calendar age had a 4-fold increased risk of dementia compared to those with the least difference. That's a lot. And certainly if I were an ApoE4 carrier, it would make me very curious to know what my biological age was. The authors also tried to explore how biological age might be related to dementia. They hypothesized that one key mediator might lie in brain volume. Maybe if we're aging faster than we should, our brain shrinks faster than it should, and that eventually leads to this thing we call dementia. MRI studies were partially supportive of this hypothesis, suggesting that about 13% of the link between biological age and dementia could be mediated by brain volume, but that leaves a lot of unknowns. But as I mentioned at the top, the really interesting thing about biological age is that unlike chronological age, it can be changed. It can go down even. Going back to that PhenoAge calculator, I can see that if I could just get my C-reactive protein down a 0.5 point, I can get my biological age down by about a year. And we don't know, of course, if changing biological age will change dementia risk. There's always the possibility that what we've seen in this study is correlation, not causation. But if calculating biological age, integrating a bunch of health metrics into one easy to understand number, is the motivational tool I need to live a little healthier, well, that might be good enough. For Medscape, I'm Perry Wilson.